to the audience so that it will be easier for them to correlate. Okay, Professor. So in the context of the stem cell research, we know the stem cells are very much similar to that of with the cancer cells. And there is huge effort to make understand how the oncogenesis happens. One of the mechanism of is that there are viruses that transform the cells into cancer cells. In that context, we have invited Professor Meir Shamay. Today he is our speaker now. Meir has started his scientific training as a master student in the laboratory of Professor Moshe Mevarek at Tel Aviv University, studying that means archaea from the Dead Sea. Then he moved to research on viruses as a graduate student in the laboratory of Professor Yosef Shaul at the Weizmann Institute of Science, and there studied carcinogenesis by hepatitis B virus. During his postdoctoral training, he joined the lab of Professor S. Diane Hayard at the Viral Oncology Program, the Johns Hopkins University School of Medicine, Baltimore, Maryland, where he studied 2 additional viruses associated with human malignancies, Kaposi sarcoma-associated herpes virus and Epstein-Barr virus. After completion of his postdoctoral training, he supervised tumor virology laboratory in the Division of Biomedical Sciences at Johns Hopkins in Singapore. In 2013, he returned to Israel to join the Faculty of Medicine in the Galilee Bar-Ilan University in Spat and establish the Viral Oncology Laboratory. The goal of his lab is to expand knowledge on viral infections and to utilize this knowledge for the development of drugs that specifically target virally infected cells. Today, Professor Meir Shamay will speak on Oncovirus manipulating endogenous transposable elements to promote tumor growth. Ladies and gentlemen, please welcome Professor Shamay.
Thank you very much for the kind introduction. You need to excuse me, I have a little cold, so I'll try to not cough too much. Okay, so in my lab, we studied the 2 herpes viruses that are associated with human malignancy, Kaposi sarcoma-associated herpes virus, known as KSHV, that is associated with all forms of Kaposi sarcoma, primary effusion lymphoma, and multicentric Castleman disease, and Epstein-Barr virus, known as EPV, which is the cause of mononucleosis, and it is associated with Burkitt lymphoma, nasopharyngeal carcinoma, Hodgkin lymphoma, post-transplant lymphoproliferative disease, diffuse large B-cell lymphoma, plasmablastic lymphoma, some of the primary effusion lymphoma, subset of T and NK cell lymphoma, and about 5 to 10% of gastric carcinoma. Like all herpes viruses, KSHV has 2 phases. One is the latency, where a few viral genomes as a big episomes, plasmids reside in the nucleus of the infected cells, and only few proteins are expressed. As opposed to that, during the lytic and replication phase, almost 100 lytic proteins are expressed, leading to the replication, assembly, and release of the virions. This release will end up by the death of the cells. As opposed to that, in latency, the cell can grow for many years, and we know that once we are infected with herpes virus, it's for life. And because many of the cancer associated with these viruses are within the latency phase, we are more interested in the latency phase. And in my lab, we ask, what is the virus is doing to the cells leading to the development of cancer? DNA methylation is an epigenetic mark associated with transcription repression, and here a cytosine is modified by a DNA methyltransferase by covalently adding a methyl group, resulting in 5-methyl cytosine. Usually throughout the human genome, we have CpG methylation. It means C followed by G. And these CPGs are not equally distributed in the genome. There are some concentrated islands known as CPG islands, and these islands are found in many promoters. Many of them are housekeeping genes, and in normal cells, these islands are protected from methylation. As opposed to that, outside the these promoters and in gene bodies, we have very sparse CPGs, and usually these CPGs are heavily methylated, hypermethylated. As opposed to that, in cancer cells, we found that some of these CPG islands becoming aberrantly methylated, the genes are repressed. Some of them are tumor suppressors. So instead of mutating the tumor suppressor gene, you just suppress its expression. Also, outside the CPG islands, along the gene bodies, we can see in many cancers there is a global hypomethylation. So when we started our project, we asked what's going on during KSHV infection. So we performed the Infinium human methylation array that has 450,000 CPGs that they can analyze CPGs in a single base resolution. And here we compare the B-job. B-job is a B-cell lymphoma, a KSHV negative B-cell lymphoma, and we compare to a PEL. Just maybe I can. Okay. So BC3 and BCBL1 are two primary effusion lymphoma, KSHV infected, and are dependent on KSHV. And we performed this global analysis. We found when you compare BC3 to B-job that 6% of the genome becoming hypermethylated, more methylated, while 30% of the genome become hypomethylated, less methylated in BC3, and very similar we found in BCBL1. So we found very dramatic hypomethylation of almost 30% of the genome. But we know this is a cancer. We know what is the contribution of the virus to this. So what we did, we took these B-job cells and we infected in the lab. We infected with a recombinant virus, 219. So these are the B-job 219 that we newly infected in the lab. What we found already following infection, and we grow the cells for a couple of weeks, we found already 4.4 hypermethylation of the genome, very similar to the 6% we found in PEL. However, the hypomethylation is very, very different. Only 2.6% of hypomethylation compared to 30%. Here we see the graph. Hypermethylation is very close. Hypomethylation is very different. This suggests that the virus can lead to hypermethylation, while maybe hypomethylation is the process of chronic infection and development of a tumor. To ask this more directly, we collaborated with the late Enrique Mesri, and he developed this mice model. He can take endothelial cells from a mice and infected them in vitro with a recombinant virus that express GFP and also have hygromycin selection. So because these plasmids don't integrate, in culture we need to add a drug to keep them in the cells. So if it doesn't add a drug, the cells lose the virus, and if we inoculated that in SCID mice, no tumor is developed. However, if it takes the infected cells, inoculate within mice, 100% of these mice will develop a tumor within 3 to 4 weeks. Now also we can take this tumor and grow it in vitro. So now in vitro, without selection, these cells lose the virus. Now we ask what happens if now we re-inoculate these cells within the mice, and now once we inoculate these preformed tumor, now we don't have the virus, but we still develop a tumor, meaning that the virus left something within the cells. And because 100% of the mice develop tumor, we don't think that random mutation is the explanation. We thought it's maybe via epigenetic signature and maybe DNA methylation. Therefore, we check the DNA methylation in this tumor compared to the cell. And we can find hypomethylation of most of these regions we look at. If we try now to look for differentially methylated promoter, we can see a very clear bias towards hypomethylation. And when we combine methylation of the promoter with expression, we can see very clear that almost all of them, 340, were hypomethylated and upregulated, and only 6% were downregulated and methylated. This suggests that during the process of development involved by the virus, we can see hypomethylation. But these are mice. Then we ask what happened within clinical samples. So we took Kaposi sarcoma biopsies, and we took from plaque, which is a less advanced disease, and nodule. And we performed the DNA methylation analysis. We found that the hypermethylation, again, is very close to each other. However, the hypomethylation is very different between plaque and nodule, suggesting that hypomethylation is associated with the development of tumor. So all these three studies led us to a model where we suggest that hypermethylation is made by the virus initially after infection, while hypomethylation is a process associated more with chronic infection and development of the tumor. And in these studies, we showed specific promoters and genes that were upregulated by this hypo and repressed by hyper. But then we thought maybe there is a bigger picture to that. How can we explain this 30% hypomethylation? And we know that one of the important roles of DNA methylation within the cell is to repress transposable elements. So if this is 100% of the human genome, less than 2% are exons that code for proteins. About 20% are line, transposable element, 13% are sign, 8% are LTR. Altogether, about 50% of our genome is are transposable elements. They are not situated like that, but they are spread throughout the genome. Some of them can serve as alternative promoter. Some sign element can serve as enhancers in gene expression and tissue development. Some of them are within introns, and it was shown that the amount and the direction of line 1 within these introns can determine gene expression. So if you have more integration of line 1 in the inverse orientation, we have lower expression of a gene. And also because they have poly A in their DNA, if they can integrate within an exon, they also can lead to alternative polyadenylation of a gene. So altogether, we can see that this transposable element really intertwine within the genome and can regulate gene expression. What are these elements? So we have the DNA transposons that occupy 3% of the genome, and this DNA transposon jump via cut and paste mechanism. While also we have the retrotransposons, they jump via copy and paste, okay? They make RNA, and then they have reverse transcriptase activity that will convert the RNA to cDNA that can integrate into new places within the genome. The retrotransposons are divided further to LTR retrotransposons, known also as ERV, endogenous retrovirus, because they are very similar to retroviruses. They have the LTR, some of them also have the ENV, GAG, POL, and 3' LTR. What we believe now is they don't jump anymore in our genome, and they occupy 8% of our genome. The non-LTR retrotransposons are divided to the long interspersed element line 1 and the short sign. Line 1 is a 6 kilobase element with 3' UTR and code for two proteins, OF1, which protein that bind RNA, and OF2 that has reverse transcriptase and endonuclease activity. Line 1 by itself occupy 17% of the human genome. There are about 500,000 copies of line 1 in the genome. However, only 100 are still what we call young and active because they are full length and they can still jump. And every newborn baby has some new insertions that he didn't have in his parents. And we believe that this maybe contribute to the evolution. Sign are very short element of 300 base pairs, and they don't code for any functional protein. Therefore, they are totally dependent on line 1 for line 1 of 2 for retrotransposition. And we have 13% of the genome are sign. So what still jump in the genome are line and sign. We are all mammals, and mammals have placenta. So how can you make placenta? Placenta is a fusion of two adjacent cells to a multicellular, and the protein that generates placenta is syncytin encoded by a retrovirus, endogenous retrovirus. So all mammals utilize different ERV syncytin to produce the placenta. During embryogenesis, line 1 is expressed, and if you inhibit line 1 expression or line 1 RT, you can inhibit embryo development. Here we can see how in different cells you have line 1 mRNA expression, line 1 of 1 protein, and a retrotransposition assay that you can do in the cells to see how much retrotransposition events occur during the experiment. So I mentioned that because they can jump, you would like in somatic cells to repress line 1, so they won't jump anymore. But in stem cells, you see very high line 1 expression. But although it's very high, you see only few retrotransposition because you have also other mechanisms to inhibit line 1 retrotransposition. In other cells, like hematopoietic stem cells, mesenchymal stem cells, or fibroblasts or keratinocytes, now line 1 is totally repressed. You can't find line 1 expression. But in NPC, neuronal progenitor cells, you can see line 1 expression, and you can see line 1 retrotransposition. And if you do analysis in single cell retrotransposition, you can see in different cells in the brain, you get different retrotransposition. Also, line 1 expression is critical for the expression of neuronal function and neuronal gene expression. Because now we're also dealing with stem cells here in this course, I brought this to you, this paper in Cell, showing that in the 2-cell state, DAX is active, and also a endogenous retrotransposable element, MERVL, is also active to keep the 2-cell state. Once you progress to blastocyte and stem cells, now line 1 is expressed. So in this study, what they did, they took the stem cells and inhibit line 1. What they found is that now now these cells go back to the two-cell state. Mechanistically, what they found is that line 1 RNA serve like a long non-coding RNA as a scaffold, bringing nucleolin with CAP1 to repress DAX expression. So if in stem cells now you repress the expression of line 1, now DAX will be re-expressed and you push the cells back to this two-cell state. Another example of transposable elements play a role in stem cells. I'm sure you are aware of the induced pluripotent cells, IPS cells. And here we can see the process of creating these IPS. We can see that nanog is expressed, but also the endogenous retrovirus HERV-H is highly upregulated, as opposed to the cells before the IPS creation. What happens if we knock down HERV-H, and we can see this is the control, and we have 4 different shRNA targeting HERV-H. We can see that HERV-H is downregulated, but now the Yamanaka factors are also downregulated. And if we now downregulate HERV-H and we check how many IPS colonies we can obtain, we can see by knocking down HERV, we can really reduce very sharply the amount of IPS cells we can create. And mechanistically, what they found is, again, this HERV-H serve as a scaffold long non-coding RNA by creating a complex on OCT4 and bringing the coactivator P300. So we can see also in IPS cells, line 1 is really critical. Now let's go back to our project. So we did RNA sequencing, and I need to mention, if you just do RNA sequencing, you will never see transposable elements, because the first step after that you do alignment to the genome is that you look for uniquely aligned region, because by their nature, transposable elements are not unique, they are repetitive, they are all thrown out of your analysis. So if you want to look for transposable element, you need to do specific different analysis. So when we did this, we found that many transposable elements were upregulated, and here I want to concentrate on the young and active line 1. We can see it is sharply upregulated in both BC3 and BCBL1 cells. To validate that, we did RT-qPCR with 3 primers for the 5' UTR, OF1 and OF2. We can see that all these 3 primers found upregulation of line 1 in the 4 PEL cell lines relative to the control lymphoma without the virus. This is on the level of RNA. We wanted to see what's going on the level of protein, and we use antibody for OF1. We can see compared to BJAB and K562, all 4 PEL cells have high level of line 1 protein. But also we wanted to check what about line 1 RT activity. So what we do here, we take the cell extract and we incubate with an RNA that we don't have in the cells with a primer. And now if we have RT activity, it will create cDNA, and therefore later on we can do PCR. So we can see negative control, we don't see anything. If we put HIV RT, this is the positive control. We can see compared to BJAB, already BJAB infected, they have almost the twice amount of RT activity, while the 2 PEL cell lines have very strong RT activity. So we can conclude that line 1 is upregulated and pale cells possess high RT activity. Also here we show the experiment for BJAB and infected BJAB. We can see upregulation of line 1, and this is the RNA sequencing, and also via RT-PCR for the general line 1 and also for some specific line 1 elements, location in the genome. So we started our project by looking at DNA methylation. Therefore, we asked whether indeed we see difference in the DNA methylation status. So we performed the bisulfite sequencing. So each row is one clone and each column is one specific CpG, and the black circles represent methylated and the empty circles represent unmethylated CpGs. Also here we have the red circles, meaning mutation, because we have so many elements, or some have some changes in their sequence. So compared to the BJAB, we can see that BC3 and BCBL1 clearly have much less DNA methylation. Here we did significant statistic of all these results, and we can see that compared to the BJAB, we can see reduction or hypomethylation of line 1 in BC3 and BCBL. This is the DNA methylation. What about the chromatin state? So we use two histone marks. One is the H3K27 trimethylation, which is a histone mark of repressive complex, repressive histone mark, and the activating histone mark, H3K27 acetylation. And we perform CHIP assay with BJAB and BC3 with a line 1 general primer. We can see more of the open chromatin. This is with the general primer, and once we go for a specific region, we can see less of the repressive mark and more of the activating mark. Also, we did the same for BJAB and BJAB 219. With the general primer, we didn't see much. However, when we look on specific line 1 region, we can see less repressive and more activating mark. This also suggests that maybe changes in chromatin, open chromatin, precedes hypomethylation. So it was shown that for some RT inhibitors, and these RT inhibitors were originally developed for HIV, but also it seems that they can inhibit line 1 RT. So you can treat cancer cells and they can stop growing. So we did the same for our pale cell lines. And we used two different RT inhibitors, efavirenz and rilpivirine, and we use three different cell lines, pale cell lines, and we can see compared to the green, as we increase the dose, we can see a dose-dependent inhibition of cell growth. Here also we compare to BJAB, so although efavirenz has some inhibition of cell growth in BJAB, rilpivirine seems to be very specific, and it doesn't inhibit cell proliferation in BJAB. However, it can efficiently inhibit cell proliferation in these three pale cells in a dose-dependent manner. We know that drugs are usually not so specific, so we also wanted to show in another way that by inhibiting line 1 expression, we can inhibit pale cell proliferation. So we use shRNA. Here we use shRNA. We validated knockdown of line 1. And here, by the proliferation, we can see that this shRNA strongly inhibited pale cell growth. Also, we did the same for siRNA, and we see the same inhibition of pale cell growth. So we can conclude that line 1 contribute to cell pale proliferation. So how come treating already a cancer cell with RT inhibitor can have effect? Because if everything is retrotransposition, the cancer cell already have this retrotransposition. So what we believe is transposition independent effect, and more change in gene expression. Let me remind you a little bit on microRNA. MicroRNA are made by RNA polymerase II as a long RNA. It has as a prime microRNA with a stem loop, and Drosha recognized this structure and then make a shorter element, and these microRNA are getting out of the nucleus, and in the cytoplasm it is digested by dicer to a 20 nucleotides that now will incorporate into the RIS complex. And if the sequence is very similar all the way homologous to the target, you will get degradation of the target RNA. But if there is some mismatches, we will not get the degradation, but more inhibition of translation. So usually, as I mentioned, microRNA are made from a pre-microRNA that create this stem and loop that process to pre and a mature microRNA. However, in cells that express high RT activity, this RNA is converted to a RNA-DNA hybrid. And this RNA-DNA hybrid cannot create this stem and loop, and therefore cannot be processed to a mature microRNA, and then we can get disruption of the microRNA pool. When we treat the cells with a Faveres RT inhibitor, now we inhibit the RT, and now we can retain the normal microRNA pool, and we know that microRNA are very important for fine-tuning of gene expression. So here we ask, what happened to the microRNA? We did microRNA sequencing following treatment with a Faveres, and we have 3 biological replicates. These are cellular microRNAs, and we can see that many microRNAs now are upregulated, and only few are downregulated, one we treat with a Faveres. KSFG also codes for about 24 microRNAs, but it seems these microRNAs are not sensitive to this disturbation by a Faveres. Actually, it was suggested that microRNA that found within introns together with SINE are more prone to this cDNA creation, because SINE have a way to attract the RT activity, and these microRNA will be more prone to the effect by line 1 RT. Here we validated 2 of these, one upregulated and one downregulated microRNA. Previously, we have done RNA sequencing comparing BJAB with BCBL1, and we identified 1600 genes were upregulated and 1500 genes that are downregulated. Now we ask, what happens if we now do RNA sequencing on this pair following treatment with a Faveres? We have found 885 genes downregulated, and actually 120 of the upregulated genes in PAL actually will be downregulated once we treat with a Faveres. Also, we found that among these downregulated genes, some can be upregulated once we treat with a Faveres. And all this project we started because we thought maybe some of the gene expression we see in PAL is actually not attributed directly to the virus, but indirectly the virus upregulate line 1, and line 1 make these gene expression changes. We also performed the microRNA sequencing, and we looked for microRNA sequencing that are up that potentially have a seed sequence target in the genes that are down with the Faveres. And we identify 123 such genes. And also we looked on microRNA that are down that can target genes that are up with the Faveres, and we identified 27 such. Now we took these lists and we performed gene ontology analysis. With the list of downregulated genes targets by upregulated microRNA, We found many pathways, and all these pathways in orange actually are neuronal specific pathways. When we ask the gene ontology analysis, what is the tissue of these genes, we can see the upper two hits are cerebellum and brain, suggesting that many of the genes that usually are upregulated in PEL and now downregulated because of a Favirez are actually upregulated because of line 1. When we look at the genes that are upregulated genes target but downregulated microRNA, we couldn't find something striking. These are 5 transcription factors that are essential for neuronal development. We can see out of these 5 transcription factors, only 1 is not upregulated. All the rest are highly upregulated in PEL. Okay, in agreement with maybe differentiation tumor towards neuronal gene expression. And I want to remind you that these are B-cell lymphoma that now express neuronal genes. To show more directly that indeed the repression is via the microRNA, we took one example, this gene TUBB3, and once we clone this in a plasmid, we found that treating with a Favirez leads to low expression of this gene. To prove that the 3' UTR is important, we took the luciferase reporter gene, and now we treated with the Favirez. We didn't see any change in expression. However, once we inserted downstream to the luciferase the 3' UTR of this gene, now when we treated with the Favirez, we got some repression of expression. Okay, now let's try to move a little bit from these B cells to epithelial cells. What we found is that once we infected, these are the magenta are the infected, MCF10A are epithelial cells not transformed. We didn't see any upregulation of line 1. When we grew these cells in 2D culture, when we grew them on the plastic plate. However, usually cells doesn't sit on plastic plate. So if you generate a spheroid and you put these spheroids on extracellular matrix or fibrin, then in this 3D culture, we can see very strong upregulation of line 1, specifically in the KSHV infected cells. And I need to mention that it was puzzling why when we infect cells in 2D, we don't see any advantage in the growth when we infect cells with KSHV. However, it was shown that putting these cells in 3D culture, now these cells infected make sprouting and have this tumorigenic process of endothelial to mesenchymal transition, EMT. So we ask what happens now with the line 1 inhibitor. First, we can see that uninfected cells and infected cells, we can see in the infected cell we have much more sprouting, and here we quantify much more sprouting. And now we take these infected cells, treat with the vehicle DMSO or a Favirez RT inhibitor. We can see that in a Favirez treated cells, we can see much less of the sprouting, and here we quantify. Also, we wanted to follow the EMT. We used 2 markers of EMT, ZEB1 and SLUG, and we can find that while infecting the cells lead to upregulation of this EMT marker, once we treat with a Favirez, this increase is inhibited. So we conclude that inhibition of line 1 inhibit this transformation in 3D, the sprouting and the EMT. So in conclusion, we have tested several RT inhibitors. We found that the Favirez can inhibit PEL cell proliferation. However, it was not meant for line 1, and we need very high concentration that we cannot achieve in the blood. But in the case of relpivirine, we find that they inhibit pale cell proliferation, and it was shown to be safe. This concentration was shown to be safe in clinical trial. Usually when we treat AIDS patients, we use a lower concentration, which is sufficient for HIV-RT. But here, what we suggest, maybe we can increase the concentration of some of the RT inhibitors in order to treat patients with pale NKS. So we are coming to the model that KSHV upregulate line 1. We have more RT activity. This RT activity leads to RNA-DNA hybrid, and these RNA-DNA hybrid dysregulate the microRNA pool, leads to upregulation of neuronal genes, loss of cell identity, and enhanced cell proliferation. And once we treat with RT inhibitor, we can reverse this path. So I would like to thank to the students, Anuj Ahuja, Guy Juno, Supriya Bhattacharya, Ron Eitan, and Boris Rapoport that work on this project and the funding.
Thank you very much, Meir. And now we can ask, you can interact with him. Anybody has got any question? I would ask a question, Meir. Myself, Sujoy, and I will ask a question to you. So where does it lead to? What is the big picture of when you talk about the oncoviruses play the role in the cancer development, what is the bigger picture? I just wanted to know.
So I didn't mention, but also in the case of hepatitis B virus and also HCV, you can see upregulation of line 1 and more retrotransposition events. And maybe we can think that the global hypomethylation we see in many cancer will lead to upregulation of line 1 and change in gene expression or lose of what we understand about cancer is lose differentiation. You de-differentiate. And upregulating line 1 leads to this de-differentiation. So maybe also other viruses are doing the same, and this might lead to the proliferation of the tumor.
So when you talk about in the context of stem cells, for example, embryonic stem cells or iPS cells, where we find that line 1 has got a expression of the line 1 is higher. So is it the stage specific development? Is it a stage specific expression like when?
Yes, I didn't get to it. So usually at the 2 cell state, it's very low. Then 6 and 8, it's coming very high, and then it gets lower again. So there is a very specific when it is high, and then it needs to go down. It was a Nature paper showing that if you force overexpression of line 1, you cannot proceed through more than the 8-day cell state.
Is there any equivalent state in during transformation from a normal cells, let's say EMT, let's say, is there any such equivalent state wherein we have a similar situations of the mimicking the stem cell situations of the expression of line 1? See, in other words, let's say there is an EMT and we know the data from the stem cells. And now, is there any overlapping situations in which we can mimic, we can think of the normal sense cells undergoing oncogenesis are mimicking the line 1? No.
So maybe I need to say that in people talking about EMT, it's not really EMT. So if you have the epithelial and you have the mesenchymal, so within cancer cells, you don't have only mesenchymal, but you only don't have the epithelial. The cells stuck in the middle. It's a transition state between real epithelial and real mesenchymal. And this seems very, because also you can find mesenchymal cells that now going the inverse direction, but usually it will not go all the way. It will stuck in the middle of less differentiated. And maybe what we suggest, maybe line 1 by leading to the differentiating maybe contribute to this process.
Okay, there was a question from the online someone. If you can address that, please.
I see. Can you discuss any recent advancement in our understanding of KSHV tumorigenesis, therapeutic potential in? So still we don't have any really good drug against tumors by KSHV. PEL cells is very, the survival with PEL is very poor. Maybe the median is 6 months and there is no really cure. For KSHV, treating the patient with HIV cocktail and by bringing back his immune system, you can really decrease the incidence of KS. Although it's not zero, there are some patients that even once you treat them with RT inhibitors and they are still having a KS and PEL, but they are a minority.
Okay. Yeah, go ahead, please.
So I have two questions for you. So this is Dr. Ghosh. I'm a colleague of Dr. Dhada. So I have two questions. One is like if you downregulate line 1, it goes to the stage of 2 cell stage of the embryo. Is it? Now, do we have a biochemical inhibitor which can downregulate line 1?
So people are using both shRNA, siRNA, and also antisense oligo against line 1. Or the other way is to do a RT inhibitor, although RT inhibitor will not inhibit this formation of the line 1 will be still expressed. So at least as a scaffold, it can still serve and it still make the protein, but RT will not be there. So I think it's a very interesting field for further research is how can you really inhibit the line 1 in the case, in the incidence of cancer.
And second question I have, like your transposon element. So by doing a normal, your global genome analysis, you cannot detect that signal. You said that you did a different kind of analysis. What is that analysis you did?
So there is a site called the rep base. So what you do, you don't align to the genome, but you only align to all the elements. You align only to the elements. Therefore, you can quantify how much reads you can align to specific elements, and by that you can compare the relative expression of the transposable elements.
Okay. So, is anybody has got any question for him? Yeah, Rashmi, come forward. Yeah. There is a student who is going to ask you a question.
Sure.
Sir, did you use any particular softwares to check the upregulation of microRNAs?
Yes, but for that I didn't get too much into that. My bioinformatician did the proper analysis for microRNA. There are already made the pipelines to analyze the expression of microRNAs.
Yes, sir.
Okay.
Thank you.
Okay, so other day we have heard Supriya Bhattacharya. It was a fantastic talk. He has done a wonderful job, and we really appreciate your supervision. And we would like to see much more work from your laboratory on this. At least it was a sort of, for us, a sort of he has made a bridge between us and you, your lab at this point. So I would like to thank on behalf of everybody from this hall as well as those who have joined from online. We would like to thank you very much for taking your time despite of having a little bit of cold and other things. Still, you have managed to give a talk to us. Thank you so much. Let's give a big applause for it from